Okay, today we're going to talk about the cell cycle, and the cell cycle is the sequence of growth and division of a cell. It has three main stages. The first stage is interphase. The second main stage is mitosis, and mitosis is divided into four different parts, and then we have cytokinesis. The first phase, interphase, is where the cell spends most of its life. And so you can see here from this diagram that interphase is actually going to be this entire circle of red here. Everything that is surrounded by the red arrow. So there's three parts to interphase, but this is the entire thing. So you can see that the cell is going to spend most of its life cycle in that particular phase. Interphase, after interphase, then you're going to have mitosis. Mitosis is this little yellow shaped wedge right there, and again it's divided into four sections because it's a cell division, but it's actually a very short period of the cell's life. After that, the third main stage is going to be cytokinesis, and that's represented by this purple wedge here. And cytokinesis is where the cytoplasm divides, the cells completely divide. You end up with your two new daughter cells from the original parent cell. So let's talk about interphase first, since that's the first phase and it's the one that the cell spends most of its life in. Interphase has a lot of stuff going on. In the first part of interphase, the cell is actually going to grow and get bigger. There is a limit to its size, but it's going to start off smaller and get larger. It's going to do its job. Whatever its job is for that organism, it's going to just live and do its job. Then it's going to work on DNA replication. So it's going to be dividing into two new cells during mitosis. And so each of those two new cells need the DNA because the DNA is the instructions for those cells. So to be able to know what they're supposed to do, what size, how long they're supposed to live, all of the things that those cells need is going to be in the DNA instructions. So it has to duplicate that, double that, so each cell has a copy. Towards the end of interphase, again preparing for mitosis to start, the centrioles are going to replicate. Oops, we don't want to go to that yet. The centrioles are going to replicate. The cell's going to prepare for cell division. The centrioles, we'll talk about those here in a little bit. They're going to be helpful for pulling the cell apart into two new cells. All right, so we have 6 different stages that we're going to learn about. Interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. Just remember that prophase, metaphase, anaphase, and telophase, those are the steps of mitosis when the cell is dividing, which is what mitosis is. Interphase is before, cytokinesis is the cytoplasm dividing afterwards. All right, we already said that during interphase that's where the cell spends most of its life cycle. It occurs before mitosis or cell division. We said the chromosomes are going to be copied, so it's going to double the number of chromosomes. Again, the chromosomes are the DNA, so that's the instructions that the cell is going to need. And at this time, the chromosomes don't look like those little X's. They're going to kind of look like the stringy stuff in the cell, and it's called chromatin. So you have these thread-like coils that are chromatin at the start, but then each chromosome is going to copy, and so it's going to have its sister chromosome. And then towards the end of this phase, they're going to change into chromatids. So they're going to condense, that DNA is going to condense into those little X-shaped chromatids. Here's a picture of interphase in both a plant cell and an animal cell. So you can see the nucleus. You can see there's a nuclear membrane that's containing the nucleus. In the next stage, in prophase, that's the first part of mitosis, the first stage of cell division, the cell is going to start dividing. And this is the point where the chromosomes really become visible. So instead of having that stringy chromatin, boy, that really keeps going to the next slide on me. The chromosomes aren't the chromatin anymore. They start wrapping up and condensing into the X-shaped chromosomes. So at this point, you can see the chromosomes under a microscope. The nuclear membrane is going to disappear. So this right here that's containing the DNA and it's keeping it in the nucleus is going to start to go away. It needs to go away so that those chromosomes can move throughout the cell and not be stuck inside the nucleus. You're also going to get centrioles. Those are these little structures right here. They kind of look like little barrels on a diagram. Those are going to move to the opposite ends of the cell, and you get what are called spindle fibers forming between the poles. So these centrioles are going to move to the ends, which are going to be the poles. Spindle fibers are going to start coming out from them. You can kind of see the little fibers right here on this diagram, and those are going to attach to the chromosomes to help move them around the cell. Here's a picture of what an animal cell and a plant cell look like during prophase. You can see chromosomes are starting to condense and form, spindle fibers are forming, and the centrioles are going to move to the opposite ends of the cell. The next phase, the third phase, is metaphase, and that is where the chromatids line up in the middle. So you can see here in both of these diagrams of an animal cell and a plant cell that the chromosomes or chromatids, the pairs of chromosomes, are going to line up in the middle. They are now attached to the spindle fiber. So those little fibers are coming out, they're hooking onto the centromere, that little circle in the middle, and they're going to be used, those spindle fibers are going to work to pull apart those chromosomes. In the next stage, here's our picture of metaphase in both a plant cell and an animal cell. You can see the chromosomes there lined up in the middle of the cell. Here you can really see that spindle attached to them and the centromeres that are going to be working to pull those chromosomes apart, because that's what happens in anaphase. In anaphase, the third part of cell division, the chromatids are going to start to separate. They're going to get pulled to opposite ends of the cell. So here they separate into individual chromatids. They're not those double X's anymore. The spindle fiber and the centrioles are going to work to just pull them apart to opposite ends. Here's what anaphase looks like in a plant and an animal cell. There we go. So you can see the chromosomes being pulled to either end by the spindle fibers and the centrioles. And then the last part of cell division is called telophase. And during telophase, this is when the nuclear membrane, this membrane that's going to contain the nucleus, is going to start to come back. And so you can see here it's reforming around the chromosomes, and now there are two nuclei. There's a nucleus here and a nucleus here. And then towards the end of this, the chromosomes are going to go back into chromatid. So until at this point they've kind of been bundled up so that they're easier to move around the cell and they can get to where they need to be. Now they're where they need to be, and they're going to kind of start to unwind back into that chromatin. And this is the end of cell division. Telophase is the last step of mitosis. So here you can see telophase in both an animal and a plant cell. And then the last stage of the cell cycle, it's not actually part of cell division. It occurs after mitosis, which is what cell division is. The cytoplasm, the stuff that fills the cell, is going to finish dividing. The cell membrane is going to move in more and completely pinch together and separate those two cells into two completely new cells, each with their own nucleus, and they have genetically identical chromosomes. So they're exact copies of each other. The DNA in this cell is exactly the same as the DNA in this cell. At this point, the cells, the new cells, are going to go back through the cell cycle. They're going to return to interphase and go through the entire cell process again.